Hello, everyone, and welcome to today's webinar, Revolutionizing Drug Development, Synthetic Nanodiscs, Unlock the Potential of Undruggable Membrane Proteins. I am Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by DEMA Biotech. To learn more, visit www.demabio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use that to submit any technical issues here as well if you're having trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Donghui Ma, founder and CEO of DEMA Biotechnology Ltd. Dr. Ma, welcome. You may now begin your presentation.
Hi, Cassie. Thank you for the introduction. It's a great pleasure for me to present DEMA Biotech's new technology platform called DEMPro. This DEMPro technology is focusing on the multi-pass transmembrane protein technology platform development. And in today's talk, we, even though we have a lot of different technology platforms for multi-pass transmembrane proteins, today we'll be more focused, elucidate more in detail on the synthetic nanodisc technology solution for transmembrane proteins. In today's talk, I will divide our presentation in 4 sections. The first part, I will talk about membrane proteins, why there are so many technical challenges for membrane protein. There's intrinsic issues with transmembrane protein because they are highly hydrophobic. And the second part will go to our today's major focus on the nanodisc. What is a nanodisc? How can nanodisc help for the basic research science and biomedical research on transmembrane proteins, and how can they utilize for different applications? And the third part, I'm talking about the preparation method for nanodisc. Currently, there are 2 different ways: MSP-based, the synthetic nanopolymer, synthetic polymer-based approach, and also DEMA's off-the-shelf product collections. On the third session, we'll talk about DEMA's other technology platform for transmembrane protein development. And last and most importantly, how DEMA, we can help customer for their technical problems, how we can provide our service solutions for the customer. And the membrane proteins, which are, people always say is a crown of the crown for the protein research, because they are the toughest group of protein to do research. And the membrane protein, based on how they attach to the membrane, it can be grouped into 3 different categories: the integral, lipid-anchored membrane protein, and the peripheral membrane protein. Today, we'll be more focused on the transmembrane protein, which are a group of integral proteins. This group of protein include receptor ion channel transporters and signal transducers. They are a group of protein which communicate between the extracellular stimuli and the intracellular signal transduction pathway. So they are the upfront communicated with extra environment. Membrane protein almost account for 25% of the whole human proteome. So they are the most important group of protein in the signal transduction pathway. In terms of the drug development, membrane protein are also important. From all so far FDA-approved small molecule and antibody-based drug, they account for almost 60% of all the drug targets. If you account for antibody-based drug targets, they account for more than 90% of that. Another 10% are the secreted molecule. So membrane protein are highly important, but they are also very hard and tough to study. Why there are a lot of technical challenges for studying membrane protein? First, the protein yield, when you make membrane protein, they tend to give a very low yield because their distribution only on the very thin level on the cell surface. So the protein yield is low. The second most challenge is membrane protein has a very unique structure. So extracellular and intracellular, they are hydrophilic, but they do have a portion called the transmembrane part. This part is highly hydrophobic. They were embedded in the lipid bilayer. So when you purify in the aqueous solution, they tend to interact with each other, form an aggregate, and lose their function during the aggregation process. So it's very hard to make those membrane protein in the biophysical aqueous solution. So it's very hard to study them. Right now, due to the technical challenge, only 0.5% of the PDB structure are membrane protein. So you can see how tough. Another thing is membrane protein, even though they are the important drug targets, like a GPCR protein, they are like more than 800 of them. They are all important drug targets, but so far, only 3 FDA-approved antibody-based drug for GPCR proteins. Recently, there's another one, GPRC5D, which target on the multiple myeloma, being approved for a bispecific antibody from J&J in this year, August. And for membrane protein, so far they are very important. So DMA has a different technology solution to help customer to work on membrane protein for the drug development or for basic research. Based on our current technology solution, we group the membrane protein into two technical solutions. One is we prepare those membrane protein in the non-lipid aqueous solution. So this technology we use is called ECD fusion approach. This mostly for those single transmembrane protein because the extracellular part can form as a distinct protein domain. So we normally, when we produce them, we fuse with a HEAth or FC. Then we produce in the like a mammalian cell and secrete it into the media and purify them. So this is called a non-lipid bilayer environment membrane protein preparation technology. Another group of the technology we use for the so all the membrane protein, they are still full length, contain the transmembrane domain. So we have 4 different technical solution. First is called a virus-like particle solution. Another is exosome. And the third one is developed by DMA Biotech called a membrane nanoparticle, MSP technology. The fourth one, which I will talk more in detail today, is a Nanodisc technology platform. For all these proteins to ensure they are still functional, can be utilized for the drug development, we normally will characterize them after purifying them. We characterize them by the physical interaction with their ligand or receptor, if they do have a ligand or receptor. If they do not, we will characterize them by using neutralizing antibody, which are, if they were FDA-approved drug or any like a therapeutic antibody in clinical trial. So sometimes we also characterize those membrane protein by using cell-based assay. So Nanodisc technology solution. So this technology, basically we purify the multi-transmembrane proteins. They are still in the lipid bilayer environment. What is a nanodisc? Why it matters for membrane protein research? Nanodisc initially, this technology developed by a professor in University of Illinois, Urbana-Champaign, Dr. Sliga and his colleague. And initially when they noticed an interesting phenomenon. When they incubate the sera with a membrane prep, they found the membrane become disassembled into a small disc-like structure. And this disc-like structure in the center, they are still in the lipid bilayer. and the outside was circulated by a group of protein called a high-density lipoprotein, normally ApoA1. So this protein on the one side is a bipolar. So one side is hydrophobic, another side is hydrophilic. So in the center, they also embedded a transmembrane protein. The extracellular and the intracellular side face to the aqueous solution, and the transmembrane part is still embedded in the lipid bilayer. So by this way, the protein become highly stable and water soluble because they are only 5 to 15 nanometer in diameter. So you can use for different biomedical, biophysical research. And sometimes people get a little bit confused. They think Nanodisc is another reagent as a detergent. Actually, they are quite different. Detergent is also a bipolar sort of a reagent. So it's called an amphiphilic molecule. So on one side is polar, another side is hydrophobic. And the hydrophobic side will face into the like a transmembrane domain. So they encircle the protein, and the polar, hydrophilic side face into the water. So they form a micelle. And they don't contain the detergent solubilized membrane protein. They do not contain any lipid bilayer. So they are totally different. And so sometimes when the protein solubilized in detergent, they might not work properly as a native status as a nanodisc. That's why the nanodisc show some superior performance, especially for the membrane proteins. And there's another question people always ask for nanodisc, whether can they solubilize different biological membranes. So in the past decades, Sliga and his group did a couple of research. They noticed nanodisc can effectively solubilize and prepare membrane protein in a very efficient way. So in this paper, his group did a solubilization study by using nanodisc technology to study E. coli's membrane proteome. So they found so on the right side, from this SDS page analysis, you can see more than 80 to 90% of the membrane protein, they can all effectively prepare in the nanodisc solution. And they are as effective as a detergent. And also, they are protein which are like can cover by nanodisc, but do not solubilize by detergent. There are also proteins solubilized by detergent, but not covered by the nanodisc. So it's a very good solution for this E. coli membrane prep. And also, they study some of the other important biological membrane, like synaptic membrane. Synaptic membrane is a very tough membrane. They contain a lot of protein. It's a highly proteus, proteins containing membranes. They can contain presynaptic and postsynaptic membrane. So to study this membrane, they did a nanodisc preparation, and they found most of the more than 80% of the synaptic membrane also can be solubilized and prepared in nanodisc format. On the right side, you can see most of the important molecule proteins, they all solubilize and effectively by nanodisc technology. So from this previous pioneer study by Signa's group, you can see the Sniga's group, you can see nanodisc is a very effective reagent work for the membrane proteins, full-length transmembrane proteins. And for those membrane protein prepared in nanodisc, what are those applications people can utilize for biological research and biomedicine study? So here I give some examples. First, in the past, you can, in my previous slide, I already said, I mean, in the past, when people study protein structure, membrane protein is really hard to study because they are highly hydrophobic, cannot solubilize in the aqueous solution. And in the past, people always use crystallography to study structure. But right now, with a new technology, especially cryo-EM, people can now study membrane protein structure with cryo-EM technology. So with Nanodisc, people can flash freeze this protein and use cryo-EM technology to solve the structure in the most native, like a lipid bilayer environment. Nanodisc can also used for like a drug delivery. For some of the protein, they may not be stable in the aqueous solution, so they can always like pack in the nanodisc format and delivered for the animal study or clinical study. And also nanodisc can be used for therapeutic drug screening, especially for some of the like GPCR protein. In the past, it's very hard to prepare them in the active form. So by using Nanodisc, you can purify those GPCRs and also in the lipid bilayer, then you can screen for the small molecule or antibody-based drug by using this for this approach. And Nanodisc can also to utilize for the protein and lipid interaction. In this case, there's published a paper, people use FRAT technology to study how membrane protein interact with their lipid environment. Membrane protein also is, a nanodisc also can be utilized for the biopharm company to study the therapeutic antibody, how they bind to, what is the binding kinetics with their targets. So you can use nanodisc to study on the beer core or SPR. You can coat the membrane nanodisc on the beer core's golden plate, and then flow with the antibody to measure the on and off rate. Membrane protein in nanodisc can also utilize for the antibody screening, for Fiji library screening also. Membrane protein to prepare in a nanodisc in the past several decades, people have been like a study and also to use different solution. So there's a traditional MSP based, we call it also just I mentioned before, ApoA1 high density protein based nanodisc preparation, or recently developed by the synthetic polymer based. The protein based nanodisc, MSP based nanodisc technology is pretty lengthy and involve a long process. First, the protein people have to solubilize with detergent. Then they have to remove the detergent with like a BioBeads, remove the detergent and replace the detergent with artificial lipid and also MSP encircled disc. So by this way, it's a lengthy process. If your protein doesn't solubilize in a particular detergent, then you won't be able to make nanodisc. The newly developed synthetic polymer-based nanodisc is pretty straightforward. So you can, this synthetic polymer has a similar property like ApoA1 or like this MSP protein, one side hydrophobic and another hydrophilic. So they can directly incubate with membrane prep and form a nanodisc. So by this way, you can directly use this like a native membrane to prepare the nanodisc. And also it's a pretty short process, so you can keep the protein in a much more native environment. So this is an example, DMA prepared our synthetic nanodisc versus a traditional MSP-based nanodisc. On the left side is the DMA's synthetic nanodisc we prepare for 4 transmembrane protein Claudin 6. So from this SDS page gel, you can see they were purified to a high homogeneity, just a 1 band. and they can be utilized for the ELISA coating and do the ELISA analysis. And on the right side is a traditional MSP-based nanodisc technology. So you can see this I adapted from a paper published in JPC. So you can see the protein can purify to high homogeneity on the upper band QR protein, but also contain large amount of MSP protein at around between 25 kilodalton and 37 kilodalton. So if this traditional nanodisc protein, if you utilize for the as an immunogen to develop antibody, they tend to give a background, or even during the screening give a background. So synthetic nanodisc have a unique a better property for this case. If you come, here is a summary between synthetic nanodisc and MSP-based nanodisc. First, synthetic nanodisc use synthetic polymer, and MSP use the protein base, and the synthetic nanodisc use a native cell membrane, and MSP normally you have to use the artificial phospholipid. So this is two major difference. Another is synthetic nanodisc, you don't need to involve detergent in this preparation approach. So in the next couple of slides, we're going to showcase DMARS off-the-shelf nanodisc collection, and we'll showcase some of the GPCRs and some Claudin series proteins, how we prepare them. And so this is a nanodisc prepared by our synthetic nanodisc for Claudin 18.2, which are important new druggable targets. People develop an antibody for gastric cancer. And on the left side, you can see we purify to a high homogeneity. On the right side, because this is a full transmembrane domain, so you can't make an extracellular ECD domain to study because they form a two loop outside. So you have to use a full length to measure the therapeutic antibody and the protein interaction by SPR vehicle interaction. So we purify this protein in nanodisc, and we measure the kinetics with right now in phase 3 clinical trial therapeutic antibody robe-2 and you can see it shows a very good binding affinity. This is another nanodisc prepared by our technology. It's a GPRC5D, recently approved by FDA, J&J's drug, so tacrolitamab. And this nanodisc we show to purify to a high homogeneity, and also we show here with a therapeutic antibody can bind effectively. And here is another case we purify by our synthetic nanodisc. We can purify to a high homogeneity. It's a 12 transmembrane domain protein. It's a MDR1 protein. It shows it's over 100 kilodalton. It shows one band, and also can coat on ELISA plate, bind to the tag antibodies. Because a membrane protein, if people study membrane protein, they know, they are highly unstable, even during this, a shipment and the storage. To ensure what all the protein we ship to the customer, they are stable and during storage and also shipment. So we study the formulation on this nanodisc products. So right now, all the DMARS nanodisc product we offer in lyophilized form. So you can ship, we can ship them in an ambient temperature, and we did some accelerated stability test. Here, one of the tests we did with CD63 is a 4 transmembrane protein, and after we make them, so we store them in -20, and also we did a 37-degree accelerated stability test on one day 2 days and 3 days and store them in this lyophilized form in 37-degree and test. After this time, then we solubilize. We reconsider them in the water and test with the antibody and see whether they still can bind to their therapeutic antibodies. From here, you can see after the long-term storage, even in 37-degree, they are still highly stable. So currently, DMA Biotech already collect more than 400 nanodisc off-the-shelf protein. These nanodisc, like a therapeutic target protein, they can be delivered. Most of them delivered next day. Yeah, most of them we focus on the GPCRs. And besides the nanodisc technology, synthetic nanodisc prepared protein, DMA also offer other like a technology solution for transmembrane proteins. So this include like ECD protein. This especially work for those single transmembrane protein because they can form a distinct domain and easy to handle for immunization and the screening process. So for single transmembrane proteins, currently we offer more than 1,000 therapeutic target proteins. So we produce them in the H3K293 cell and human protein produced in the human cell line and purified to a high homogeneity. And to ensure the binding, we as I mentioned, we do a receptor ligand interaction if they do have a receptor ligand, and if they don't, we do the biosimilar benchmark antibody binding to ensure they are in the right conformation, native conformation. So for multi-transmembrane protein, sometimes you can't make extracellular domain, ECD domain. We also provide 3 different technology solutions to help customer to make them in native environment. So the first technology developed by DMA Biotech is a proprietary technology by DMA Biotech. So we prepare the membrane protein in a membrane nanoparticle format. We call it MNP technology. So this technology is very simple. So we first express the multi-transmembrane protein on the cell surface. Then we make a membrane prep. Then use use a physical squeezing technology to squeeze this membrane into nanovesicles. This nanovesicle normally 100 to 150 nanometer in diameter. So they are totally water soluble, and they are also highly stable. And the membrane protein is still in the membrane lipid bilayer environment. The second technology DMA offer is a traditional virus-like particle technology solution. This technology is an active secretion, bioactive secretion process. So we borrow the natural process of the virus. So the virus capsid protein, when we co-transfect with a multi-transmembrane protein, the capsid protein, they can form and like vesicles within the cell. Then they will body it out from the live cell and form a kind of like nanovesicles. This nanovesicle is around 100 to 150 nanometer in diameter. When they body it out, they also carry out cell membrane. So they carry out some of the, like if your membrane protein like distribute on membrane, they will also be passively take to this like a VLP particles. So but this technology sometimes have a little bit issue because the VLP, the capsid, when they secreted these vesicles, normally sometimes they secreted from a particular domain on the cell surface, sometimes like a lipid raft. If your protein doesn't distribute in those particular domain, so you might not be able to make VLPs. But our MMP technology is different. So we make it by different technology. So if as long as the membrane protein distribute on the like express on the cell surface, we can make as vesicles. So that's MMP and the VLP technology. The exosome is another way we make like vesicles, nanoparticle format. So that's our 3 different technology solution. So this is an example we make as an MMP. So after we make them, you can see they are water soluble. This is also Claudin 6 was an example we can use for ELISA, and also we can utilize for the flow cytometry analysis. Because the vesicle is very small, you have to use like certain flow cytometer to measure them. We call it flow varimetry. From this flow varimetry, the MMP, you can see more than 97% of the vesicles contain Claudin 6, so they distribute it in a highly ubiquitous way. And this is another example. We prepared a virus-like particle, so we utilize this technology for the CAR T cell evaluation, because after CAR T cell preparation, people always need to measure how many T cell being transduced by the lentivirus. They can express the CAR T on the cell surface. So you need to use this, membrane protein to monitor their transduction efficiency. And to ensure those different format of membrane protein, they still stable during the shipment and also storage at the customer side. We also did a formulation study, and all these a different format of the membrane protein, different preparation, we offer them in the lyophilized format. We did accelerated stable stability test. So this is a ECD membrane protein. So we did the same way. We store them after purification. We store them in -20, and also 37 degree in lyophilized form, 1 day, 2 days, and 3 days. After that, then we reconstitute and bind with therapeutic antibodies, see whether they lose activity. And this is the Claudin 18.2 VLP. We did accelerated, lyophilized forming accelerated stability test. And this is the MMP in the accelerated stability test. They are all very stable. And DMA right now we offer the largest off-the-shelf collection for full-length multi-pass transmembrane protein. Here is our collection comparing to different companies. You can see we offer the largest whole collection globally. We offer more than 480 of them off the shelf. And if some of the membrane protein, if you are interested not in the list of all the off-the-shelf products, you can always contact DMA Biotech. We do customer nanodisc preparation. So based on your needs, from the start of the gene synthesis to production to final formulation, in the lyophilized form. Thanks for today's time to attend today's talk. So here I would like to take questions. If you do have any question, do not hesitate. You can type your question in the Q&A. So I will answer the question by order. Yeah, thank you.
Thank you, Dr. Ma, for your informative presentation. As he said, we will now begin the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located in the far left of your screen. We'll answer as many of your questions as we have time for today. Our first question, Dr. Ma, says, what are the advantages of synthetic nanodisc preparation method over detergent-based preparation method for transmembrane proteins?
Yeah, this is a very good question. So as I mentioned in my talk, so detergent-based preparation approach for transmembrane protein is it's been decades. So people using this in the past successfully purify a lot of different protein. But one of the issues is the detergent. They removed the very highly native lipid bilayer environment. So they replace with like a detergent formed micelle to cover the hydrophobic transmembrane domain. So some of the so before you do the detergent solubilization of the transmembrane protein, you have to do a detergent screening. Not every detergent can solubilize a particular membrane. So, and also the environment is different. Sometimes during the detergent solubilizing, you might denature the protein. But Nanodisc is a totally native environment. The protein still embedded in the lipid bilayer. The transmembrane domain still embedded in the lipid bilayer environment. So they are highly native.
Excellent, thank you. Our next question here asks, can we make synthetic nanodisc from native cell line or tissue prep?
Yes, yeah. The nanodisc you can make you can directly add the nanodisc, synthetic nanodisc, solution to the like a native cell line membrane prep or the tissue membrane prep. Then they can directly solubilize. In the past, Dr. Sniga's group have done some of the like a pioneer research to prepare this. They call the protein library. So they study some of the like a cell, cell line prepared membrane prep for the nanodisc, and also some tissue-based membrane prep. So yeah, you can use that directly for this.
Great, thank you. Our next question here asks, can two membrane proteins integrate into one nanodisc?
Actually, how many membrane protein can integrate, can keep in one nanodisc? The nanodisc depend on the different preparation approach. Right now, the maximum like a disc people can form is normally you can reach up to 50 nanometer in diameter. So in this case, sometimes you can hold up to like 2 or 3 transmembrane protein in one disc. And also this depends on how many transmembrane domains. There are some research study say the maximum they can hold up to 36 transmembrane domains, but there's still a preliminary research. Need a more like a thorough investigation. How many transmembrane protein is the upper limit? So far, from the literature investigation, people report 2 transmembrane protein can hold up into one nanodisc. Yeah, but most of the case, they are just a single transmembrane protein in one nanodisc. That's why you can purify to a high homogeneity. I already shown in our one of the slide in my previous presentation. Yeah.
Excellent, thank you. We have another question here, and that question says, can we use nanodisc technology to study membrane protein complex?
Yes, nanodisc is a good technology to study transmembrane protein complex, because during the purification, like when we purify our nanodisc, we use a highly native environment. We don't add in any detergent, any harsh chemicals into the solution. So it's a highly native solutions. We purify them. So in this solution, you definitely can purify those transmembrane protein complex. You can study the protein interaction during the purification. You can after purify, you can run the gel, even identify those co-purified complex. And so that's one of the beauty for nanodisc. And so this is that's also during our purification. Sometimes we do observe this. So certain protein always, when you purify them, they always come along with your target protein. That's a very natural process. Yeah, that's also demonstrate your protein in the native conformation, native structure. Yeah, Cassie, thanks.
Great, thank you. All right, our next question here asks, how stable is Nanodisc solution upon reconstitution with water?
According to our study, we did some like a stability test. So after we reconstitute the lyophilized form of the Nanodisc, we leave at like a 4-degree up to a couple of months, like 2 or 3 months. They are still stable. So actually, Nanodisc is a highly stable structure. So when the transmembrane proteins stay in the Nanodisc format, they tend to be highly stable and highly water aqueous soluble.
Excellent. And it looks like we have time for one more question here today. That question asks, what is the recommended storage condition for Nanodisc?
And the recommended storage at the customer side, before reconstitute, you can store at we do all recommend that you store all your protein in at minus 80 degree. But the lyophilized form, you can, for the short time being, you can store them in minus 20, should be okay. But for long-term storage, we definitely recommend to store at a minus 80 degree. After reconstitute with the water, we recommend you to use as quick as possible. But for the short time, you can store them at a 4 degree, should be okay. Yeah.
Wonderful. Thank you again to Dr. Donghui Ma. I'd like to take this opportunity to ask if you have any final comments for our audience before we go.
Thanks for LabRoots give us this opportunity to present our Nanodisc technology. And I think a Nanodisc is a breakthrough in 2000. And even though there's a lot of publication in this area, but in the industry, there's still a lot of not many company can offer this product line. DMA Biotech, we try to be a pioneer in this area, so we understand the transmembrane protein. It is one of the major technology challenges for biopharma companies. They need this for immunization and also drug screening. So we try to provide off-the-shelf solution for the biopharma company. And if you do have any question, or if you want learn more, you can always go to our DMA Biotech's website, www.dmabio.com. So we have a lot of information for you to study and to understand different technology solution for transmembrane protein, include the Nanodisc, MMP, and also VLP. Yeah, thank you. Thanks.
Thank you, Dr. Ma, for your time today. We would also like to thank LabRoots and our sponsor, DMA Biotech, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Bye. Thank you.